Alzheimer's Treatment and Understanding by David Hahn,
Grace Kim, and Shayan Lee.
So what's so bad about having Alzheimer's disease? Let's take a look at the brain. Whoa! That does not look like a normal brain. The brain on the right is the brain of a patient who's diagnosed with Alzheimer's disease. You can tell that there is significant loss of neural tissue, and there's clear evidence of brain atrophy. Here's another comparison of a healthy brain with that of a patient who is diagnosed with Alzheimer's. Here's a neural scan of your brain if you had Alzheimer's. What is Alzheimer's disease? Alzheimer's disease is the irreversible degeneration of the brain that causes disruptions in memory, cognition, personality, and other functions that eventually lead to death from complete brain failure. Here's some quick facts. Every 60 seconds, someone in America develops Alzheimer's. It is estimated that 500,000 patients will be diagnosed this year alone. If dementia care were to equal the economy, it will become the world's 18th largest economy, coming in between Turkey and Indonesia. Did you know that the National Institute of Health funding for cancer research is 12 times the level spent for Alzheimer's disease research? But more people die from each year from Alzheimer's than from the two most commonly diagnosed types of cancer. That's right, breast and prostate cancer.
There are two phenomena that are only observed in Alzheimer patients' brains. One, an extracellular protein known as amyloid beta, a protein naturally made in the brain, forms plaques that are insoluble and unable to leave the brain, disrupting electrical signals between nerve cells. The second, tau, forms in neurons and disrupts signal and molecule transport mechanisms within the neurons. The blood-brain barrier is what protects the brain from foreign blood-borne chemicals. Specifically, tight junctions within the blood-brain barrier prevent toxic molecules from passing through. However, in Alzheimer patients, the blood-brain barrier prevents amyloid beta from leaving the brain, causing plaques to form. Plaques can be prevented from forming by destroying the proteins that form the tight junctions within the blood-brain barrier, called claudin 5 and occludin, with matrix metalloproteinases, or MMPs, and causing leakages in the blood-brain barrier. Additional protection by other layers, known as parasites and astrocytes, prevent this leakage from causing any significant damage by foreign molecules. However, there's another type of barrier called the blood-cerebrospinal fluid barrier, or the blood-CSF barrier, which serves the same function as the blood-brain barrier but lacks parasites and astrocytes, making it much more vulnerable to outside molecules.
When amyloid beta is injected in mice, the gene expression for LL6, LL1B, and TNF significantly increase. This causes the inflammation of the brain within the mice, particularly in the hippocampus, which is very crucial for memory. This is among the first regions to be affected in Alzheimer's disease.
MMPs are significantly upregulated in patients with Alzheimer's because an increase in amyloid beta is shown to increase the amount of MMPs. Figure A shows that patients with higher amounts of amyloid beta have a greater membrane permeability, while figures B, C, and E show that claudin and occludin concentrations are less than the control group in which no amyloid beta was added, indicating that MMPs increased with greater concentrations of amyloid beta, destroying claudin and occludin. This picture shows how MMPs enter the tight junction within the blood-CSF barrier and destroy claudin and occludin, creating a leakage in the membrane.
Although you may expect Alzheimer's patients to have blood-brain barrier dysfunction that allows beta amyloid to enter the brain, they actually don't. This was proven in the lab with positron emission tomography, or PET scans. In this experiment, 5 Alzheimer's patients and 5 healthy people of the same age were scanned, and there was no evidence of any intravascular tracers entering the brain. There were also no differences in blood-to-brain transport between elders and Alzheimer's patients. This showed that there were no abnormalities in the blood-brain barrier of Alzheimer's patients. But it is true that Alzheimer's patients have higher blood-CSF barrier permeability. Through many studies, it was concluded that increasing age and Alzheimer's disease are both closely related to increased blood-CSF barrier permeability. To come to this conclusion, researchers conducted an experiment using 3 groups of same-age subjects, a healthy control group, a neurological control group which consisted of people who suffered brain disorders like vertigo or chronic headache but not Alzheimer's, and a group of Alzheimer's patients. To evaluate the blood CSF barrier dysfunction in Alzheimer's patients, CSF activity of an enzyme called SPLA2 was compared across the three groups. Researchers observed higher SPLA2 activity in the neurological disorder and Alzheimer's groups compared with the healthy control group. Here, you can see data that these researchers got off their study of these three control groups. The SPLA2 activity is significantly lower for the healthy control group compared with the neurological control and Alzheimer's group. The SPLA2 activity measurements are very specific in that way for separating healthy people from people with neurological problems. The extremely low p-value of 0.0004 shows that this data is significant. Some other things you can note in these data is the fact that the tau concentration is significantly higher in the Alzheimer's patients than in the other groups. The really low p-value supports the fact that tau probably plays a large role in causing Alzheimer's. There's also a really clear difference in amyloid beta concentration, which really illustrates the effects of the fact that the permeability of the blood CSF barrier is so much higher in Alzheimer's patients than it is in people without Alzheimer's. This graph shows the data we saw on the previous slide. Activity of SPLA2 was noticeably lower for healthy patients, and this graph makes it easy to see how comparing SPLA2 activity can easily differentiate between a healthy person and someone with some type of neurological disorder. And here you can see a visual depiction related to what I said earlier. Examining SPLA2 activity separates healthy people from anyone else with a neurological condition of any kind, including Alzheimer's. So what do we know now from these experimental studies? Well, amyloid beta buildup is the main precursor of Alzheimer's, as it increases the concentration of MMPs that are linked with causing Alzheimer's. Knowing this information is important as it gives us a better understanding of the disease itself and what mechanisms actually lead to it. The SPLA2 detection experiments provide a valuable way to detect Alzheimer's, taking us one step closer to making treatment easier.
Alzheimer's disease is still not fully understood, but the main causes of it have to do with the dysfunction of regulating fluid and chemicals within the brain in order to maintain homeostasis. Today, we are trying to find ways to prevent the buildup of amyloid beta plaque and tau proteins from spreading throughout the brain within Alzheimer's subjects.